Aaron, are we? Awesome. All right, welcome everyone to the Tools and Technology Seminar Series. I think most people have signed in. If you haven't, there's a sign-in sheet. Please put your name on it. It helps with the pizza. I'll bring it to the back for anybody who hasn't gotten it. But Gil, I can say. So sign in. Great. We do have people that are viewing remotely, so I will ask if you ask questions, if you could speak up a little bit. There are microphones in the room, but some of the soft-spoken people or people who are sitting on the periphery aren't getting picked up great. So just a reminder to everyone, if you speak up, that'll be great. So that, I'll introduce today's speaker. We have Zheng Gao, and he is a grad student in statistics over at LSA.
Thank you, Marcy, for the invitation. Today I'll be talking about a little bit of piece of software that I wrote about 6 months ago on Genomewide association studies, and I assume that most of the audience is from Department of Computational Molecular Biology. So I think people are in general more familiar with the topic than people in stats. But in general, I'd like to just remind everybody what this is. The Genomewide association study is. So basically what people are doing, and this is from my understanding, that people are scanning people's genomes. They collect a large number of samples and they scan their genetic locations looking for variations in genetic composition. And they're looking for associations with the phenotype, the disease outcomes, or some kind of phenotypic traits. And then they perform marginal association tests to try to locate the set of genetic locations that are relevant for the disease or for the condition that you're interested in. So in a way, you're trying to look for a set of small set of locations among a large number of candidate locations and try to narrow down the search. But if you look in the Genomewide association study, the Wikipedia page, I think people used to know there's this trade-off between something called allele frequency and the effect size in terms of alter. How many people have you heard of this trade-off? Yes, one. Okay, two. Right. So what this means is basically saying that allele frequency on the x-axis is saying how rare a genetic variant is in the population. So if it's rarer, it's so if you have a genetic composition which is 99% of one kind and 1% of the other kind, then you have a pretty rare. But you could go even further down. You could have a rarer mutation that makes up an even smaller fraction of the population. So that's on the x-axis. Now the y-axis is the odds ratio. It's basically saying if you have the disease, so the ratio of the odds ratio of you having the disease when you have one genetic composition versus the other. So the larger this number is, the more likely you're going to have the condition if you have the variant that causes the condition. So basically, this is a measure of the effect size measured in terms of odds ratio. So what people have found in the past, and this is from a 2012 review paper, is that there's some kind of a trade-off between the allele frequency and the effect size. So in the sense that if you have really rare variants with very small effects, imagine that some mutation is very rare in the population, but at the same time it causes the probability of you contracting the disease or you developing the symptom to go up by not so much, then it's really hard to find in these kind of studies. So you found virtually nothing in these regions. But if you have very common mutations with larger effects, it's usually easier to find in these kind of screening studies. So there's this kind of diagram that people drew. But I want to draw attention to one thing. If you look at the y-axis, the y-axis goes from 1 to infinity. Okay, at what scale do you have an y-axis going from 1 to infinity? Can anybody give a guess? Sorry? Cartesian plane, yes, but do you have infinity marked somewhere in the Cartesian plane? you usually go 1, 2, 3, 4. You never reach infinity, right? So so there's one giveaway that this is not really a result that's quantitative. This is a plot that the authors drew to draw attention to the qualitative fact that there's a trade-off between these two quantities. So I, in fact, emailed the author here. I said, which, first of all, it's not the regular Cartesian plane with the regular grid, right? Then it's not the log plot either, right? Otherwise, you would have, let's say, 2 and then 4 and 8, right, at a regular interval. So this is not log scale, on log scale either. So I emailed the author. I said, what scale did you use? I said, we were just trying to illustrate the conceptual point. So this is not qualitative, quantitative result. So what I did in a way is to quantify this trade-off in this little app here. So it's exactly the same thing. So each dot here, each point here is going to tell you a genetic location that they found to be associated with the condition. And the location of this point is going to tell you the estimated allele frequency and the estimated odds ratio. So this is an estimated location of that particular genetic location in this diagram. And now this app here basically quantifies everything in the sense that if you look at this part of the diagram, it looks almost similar to what we saw before, except now this is a quantitative result. In the sense that each dot here still marks one genetic location that is associated with the condition that you're interested in. And then the X and Y locations are estimated allele frequencies and the estimated odds ratios reported in the GWAS literature. Okay. And this particular point, let me point this out to you, is found so in a particular study of estimated odds ratio this much and estimated allele frequency this much with its p-values and the name of the paper, etc. Now, what is the shades of gray? What is the dark color and the bright color? So the dark color says that under some regular conditions, that if you perform the study again, you're very not likely to discover this under this particular sample sizes. And the white region, under white regions, you will have exactly power 1. You have perfect power to rediscover this association again if you were to try to reproduce this. So this is an app that basically quantifies this trade-off. So in a way, if you have rare mutations with very low effect size, then it's difficult to discover. And if it's common and has large effect size, then it's easy to discover. Now I'm going to talk about what this is in detail. But this is an overview of this app. So it's fully interactive, allows geneticists to explore what has been reported in the literature. And also it allows you to do a power analysis. So if you want to design a study, for example, if you want to design a study to you want to determine how many samples, how many people you have to recruit in order to discover a certain effect size, then you can go through the power calculations by inputting the necessary quantities, which I'll talk about in about 5 minutes. Then this will give you an approximate number of samples that you will have to collect. So how many people you will have to reach out to collect their genetic information in order to reach the conclusion that you want. So this is just an overview of the app. Okay, let's just go into the talk. So that was a very quick overview. I'm pretty sure that nobody caught everything. So this is the full agenda of today is to tell you what this app is doing really. And talk a little bit about the software, how I built it, the tools that build this tool. I think that would be of slightly broader interest. How if you want to build a dashboard like this, how can you do it? And if we have time, we'll go into this slightly more theoretical direction on why is there a black and white? Why is there 0-1 distinction in these kind of association studies when you try to find signals in a large number of candidates? So the first thing I want to talk about is what this app is really doing. So what are we doing here? In genome-wide association studies, what typically people do is, like I said, they collect the genetic variants of each locations of a large number of people, with probably half of them having the disease, and half of them don't have the disease. And then at each genetic location, what people do is classify everybody into 4 categories. So based on whether they have the condition or they don't have the condition, they fall into one of 2 categories that are either cases or controls, right? Cases standing for the fact that they have the condition, and controls if they don't. Now, based on their genetic composition at that particular genetic location, you can also classify them into 2 categories, whether they have one particular variant of the genotype or the other genotype, right? So based on this 2x2 combination, you can classify everybody in your study. For example, if you have 1 million people, which is a really large study, then you can classify this 1 million people into 4 cells. So you can count how many people have this condition with genetic variant 1, and so on, the other 3 categories. So the 4 numbers should add up to 1 million. And what people do, geneticists do, I think, is that they try to find if the genetic composition has any association with the other marginal, which is the outcome of the phenotype. So they do kind of association test between the 2 marginals. So in a way, if you imagine that if these 2 are related, this table is going to tilt towards one diagonal versus the other. Now, if they're completely independent, the number of counts in a particular cell is just going to be the total number of subjects times the 2 marginals at that particular combination, right? So if you have nothing related, it's going to be roughly independently distributed, the columns and the rows. Now, what was I doing in the app? So typically, again, the same question. If you have these kind of studies, you want to find out how many people you have to recruit in order to reach the conclusion that you desire. Okay. Now, these are the counts of the actual outcome, of the experiment. But underlying this, if you assume a multinomial table, so if you assume random sampling from the population, then there are multinomial distributions underneath this table, right? So for each combination, there's a probability associated with each of the 4 outcomes. So these 4 numbers should add up to 1. Now, if you want to test for associations, then if they're independent, then this mu11 is going to be the marginal of variant 1 times the marginal of case, for example. So that's the probability part of it. Before that, there was randomness in the sampling. Now you have the probability table. But the first question is, what kind of test should you perform on these kind of tables? And I've seen many kinds of tests that are floating around in the literature, and people do different things, indeed. So some common tests for finding association between the 2 marginals is a chi-square test, likelihood ratio test, right? And student t-test, if you just compare the proportions of people having the disease across the different genetic composition groups. Welch's t-test, logistic regressions, etc., so that you can adjust for other factors. Now, different tests, in principle, they deliver different power, right? So if you have a different procedure to discover things, then you have different power of discovering things. Now, the question is, does it matter? Does your choice of a statistic matter in this case? And what tests do all these existing power calculators assume that are floating around in the internet? It turns out it's not that clear. That you have seen colleagues in the biostats department, for example, they publish a calculator that tells you that they calculate the power for this this procedure, but they don't tell you which one. And it's quite frustrating, in fact, to find out which ones they calculate this for, unless they're very explicit about this. Of course, the next question is, does your choice matter? If you use a power calculator that is specific, for example, to logistic regression, can you use this result if you're doing student t-test, for example, a Welch's t-test? So in principle, you can't, right? But it turns out that the choice doesn't really matter when you have really large sample sizes. That's just the asymptotic equivalent. So asymptotically, so that means when you have really large samples in your genetic association studies, the answer is no. So that's very comforting, right? Whatever you find on the internet, for example, done by Gonzalo's group in the biostats department, that calculator still works, even if your test statistic doesn't exactly match what he's calculating for. But I don't think this has been said enough in the literature. In fact, I haven't seen it. So this is very comforting in a way, so that no matter what kind of test, or these kind of likelihood ratio tests and the logistic regression, Pearson's chi-square test, two-sided Welch's test, etc. All these tests have the same asymptotic power curve. So in human language, it doesn't really matter. The calculation still applies. Now, the second question is perhaps more, I don't know, more relevant, for actionable in a way for geneticists. So imagine if you're writing a grant. So you're saying that I have to collect this number of people in order to discover certain associations. How do you reach that number? You go through a calculation, right? You go through a power calculator, like what they provide, for example, Gonzalo's group. And what they ask for in those kind of calculators is these three things: the genotype relative risks, the risk value of frequency in the general population, the disease prevalence in the general population. So that's what they all ask for. So, but in my experience, very few people know precisely what they mean. Okay, so just to recap a little bit of what they mean. And okay, so now in these kind of disease models, so-called disease models, it's slightly different from the 2x2 tabulated table that we saw earlier. So in these kind of disease models, you have 3 columns instead of 2. So just now we just counted the total number of counts of certain genetic variants in 2 columns. So there could be more, but typically there are only 2 that constitutes most of the population. But in these kind of disease models, you have 3 columns, and each of the columns tells you how many copies of a genetic variant there are in one person, because you have a pair in every single person, right? So you have 0 copies. You could have 0 or 1 or 2 copies of the disease-inducing, so-called quote-unquote genetic variant in every one person. So now you could tabulate these in a 2x3 table instead of a 2x2 table. So that's what the disease model is. Now, here's what they assume in the disease models. So you have conditional probabilities of you contracting or developing a certain condition, given that you have 0 copies. So that's the first term here. So that's pi10 divided by the sum of pi10, pi20, divided by the probability of you developing the condition if you have 1 copies, divided by the probability of you developing the condition if you have 2 copies. So that's the ratio of 3 numbers. And the disease models typically falls in 1 of 4 categories. So if you're multiplicative, then the ratio of these 3 numbers is also multiplicative, in the sense that the ratio of if you have 2 copies is GRR times more likely than if you have developing the condition is GRR times more likely than if you have 1 copy, and in turn GRR times more likely if you have 0 copies, and so on. And I want to explain how the other ones. But you see, that's a very particular parametric assumption about the disease models relating to the allele copy numbers.
What is the additive model?
Oh yes. So the additive model says that the probability is progressing like with a constant difference instead of a constant ratio, right?
Yeah.
All right, and you can stop me at any time if you have questions. Thank you. So this minus that is the same as GR minus 1, right? So that's the probability. Let's say if this is 0.1 and this is 0.2, then this is 0.3. Okay. If you're a multiplicative model, it is 0.1, 0.2, and 0.4. So that's dominant basically says if it's 0.1, 0.2, and 0.2. So if it's dominant in the sense that as long as you have one copy, you have increased probability and it stays the same. And recessive means that only if you have two copies, then you have increased probability of contracting or developing the condition. Now, the second quantity they ask for is the prevalence of the disease in the population. Now, this is important. They don't tell you it's the number for the population, right? And that, in my experience, when I ask people what number is this really, prevalence of the disease? Is it disease in the sample prevalence of the disease in the sample you collect, which is typically 50%, right? If you design a study, you want 50% conditions, 50% controls. But this number is really asking for the number in the population. Yes.
So, in order to compute the conditional probabilities, in addition to knowing the GR, you are supposed to know the mechanism. It could be different.
Right. So the question is, in order to compute the GRRs. No. What's the question? Sorry.
And your power calculator also needs you to know the mechanism of action of the disease.
Right. That's a very good point. So he's saying that in order to perform this calculation, you have to know the mechanism of the disease, in the sense that you have to know
which one.
Yes, which one of the four models it is, and you also have to have an estimate of the GRR associated with that particular model. You're absolutely right. So, in order to do that, you have to know both. If you want to go through their calculation, and I'll tell you in a minute, you don't have to do that. So that's the beautiful part. And I'm arguing against, in fact, these type of calculations, because it never will be able to precisely pin down which one of the four models it is, and you'll never have an accurate reported GRR estimate in the literature. In fact, people don't report this. People report something else. I'll tell you in a minute. So the second number they require is this prevalence in the population, like I talked about. And the third number is the risk allele frequency in the population. So P, basically how many, if you mix everything together, how many of them are the alleles that are risk associated. Now, in order to proceed with the calculation, you also have to assume Hardy-Weinberg equilibrium, which dictates that these probability tables behave something like incompatible with these number P here. So now the probability tables, pi 10 to pi 22, the 2 by 3 table, are determined jointly by these three numbers, so that you can proceed with the calculation. So that's how an existing calculator proceeds. Now, that's not all. Okay, so when you have these three numbers, you have this 2 by 3 table, but that's for the population. That's not for the samples that you collect. Imagine if you have very few cases in the population, then the sum of these three numbers will be very small, right? Close to, let's say, 1% or 0.5% in population. That's how many people in population develop the condition. But in your study, you actually oversample the cases typically if you have rare diseases. And let's say you typically do a study with 50% control and 50% cases. Then you have to adjust for that fact as well. So in the sample, in the study, you have to adjust for the fact that you oversample the cases. So you kind of, in addition to this pi 11, pi 10, pi 12s, you adjust for the fact that the phi, which is the fraction of cases in the study, is larger than the prevalence that is actually in the population. So this line is scaled up, and this second line is scaled down if you oversample the cases. Now, okay, finally, if you have this 2 by 3 table for the study, you can calculate the allele counts, the original 2 by 2 tables, okay, according to all these numbers. Now, the exact parameterization doesn't really matter. It's too complicated. Nobody remembers it. I don't. But that's how it's done. So you go through the disease model, you adjust for the sampling, oversampling, undersampling in the study, and then you have the underlying probability table for the 2 by 2 table counts that you actually observe. That's how an alternative is specified in the typical power calculator. Okay. That's what we saw earlier. So each number corresponds to the other table. Now the question is, can we skip the disease model? It seems like a long-winded process. You have to assume that certain kind of biology. You have to have the estimates for that biological model. And the answer is yes. In fact, this table, if you think about it, can be parameterized quite simply. It has only 4 numbers. You don't have to go through that. And in fact, this is parameterized by 2 quantities. The first is what people say the risk allele frequency in the control group, F, which is really, when you look at the controls, the ratio of pi to 1 over the sum of pi to 1 and pi to 2. Okay. And why are we using this? Is because this number is widely reported in the genetic literature. Okay, so that's what it is. And the other quantity is this, the odds ratio of the allele variance. And this quantity is really the cross product, the ratio of the cross products in this 2 by 2 table. So pi 1 1 times pi 2 2 divided by pi 2 1 divided by pi 1 2. So basically measures how off diagonal this table is, measures how associated these 2 marginals are. So in a way, we went through the disease model, the risk allele frequencies, the population, and the prevalence, etc., so that we can derive a 2 by 2 table, which is fully parameterized by these 2 quantities. So why do we have to go through this? And the answer is no, we don't have to, right? To go through this part in order to get to the power of your analysis. And I'm even arguing that we should, and for good reasons. First, these 2 quantities are widely available in the literature. If you recall, the app that we just saw, the numbers here, the x-axis is precisely this risk allele frequency in the control group that people estimate from their study and report in the GWAS catalogs. And the y-axis is precisely this odds ratio, the ratio of the cross product in that 2 by 2 table that people actually still report in the GWAS catalogs. So these 2 numbers are widely available as opposed to the disease models. When was the last time you see a disease model and the parameters be reported? For me, it's never. So maybe for power calculations, this is the right way to go. And then there's a robustness against human errors because people don't typically know when they go through these calculations what precisely prevalence mean, right? Does it mean in population in the study, right? People can make mistakes. And then there's a third point that this type of calculation, if it goes directly through this 2 quantities, is robust against model misclassification. This goes back to the question just now. You have to know the disease. You have to know the biology. In a way, what if the ratio of the 3 probabilities is not 1, 2, and 4? What if it's 1, 3, and 4? It's not multiplicative. It's not additive. It's not dominant. It's not recessive, right? If the biology dictates that it's 1 to 3 to 4, then it doesn't fall in any of the 4 typical models that you have. So the power calculations, if you go through the disease models, won't allow you to do this. But it still allows you to go through directly the F and R calculation to get to the power. So, and then there's a compatibility issue of parameters. In disease models, not all numbers are compatible. So you can have you can have 3 numbers that are not compatible with each other. I won't go into details on that, but it could be biologically impossible if you have any arbitrary 3 number combinations in disease models. On the contrary, if you go through this, the risk allele frequency and control group and the odds ratio, any valid numbers, any number between 0 and 1 here and 0 to infinity here will be a valid pair. So there you have another layer of reduced chance of human error. And finally, and I think this is really the most important point, is that it allows you to perform systematic reviews and unified power analysis across all studies because it's independent. First of all, the first 2 slides I talked about how the test statistic doesn't really matter asymptotically, And now I talked about how the disease model can be bypassed, right, to a number that's universal to all the numbers, to all the models. So that's what I was really doing. And if this weren't very interesting, because it's just purely some kind of algebraic work and a little bit of rethinking of what people have been doing in the past. Now I'm going to talk about again the software. So now that we understand what the x-axis is and what y-axis is, so what each point, again, like I said, is at association discovered in one particular study. Now the orange point here is all the associations discovered in one particular study in 2017, and all the points are the discovered association for the disease breast cancer. So this particular study has 76,000 cases and 63,000 controls. So if you have this sample size, the power calculator predicts that you have power close to 1 in the white regions and power close to 0 in the dark regions. And when you overlay all the discoveries in that particular study, it turns out that all the discoveries lie precisely in the region where you have power to discover things. So this is an indication that this power calculation is really precise in the sense that it predicts exactly where these. This is not line fitting. This so-called phase transition of dark and light is not fitting. Is not trying to fit to the discoveries of this particular study. This is really predicting what you should be discovering, and this precisely predicts what this study has discovered. Now, if you have smaller sample sizes, for example, if you have another study, okay, so some studies don't report their number of controls, so that you get into trouble trying to calculate the power curves there. But yeah, the software is smart enough to detect that. But if you have a study with, let's say, smaller sample sizes, then you would predict a smaller region of discovery, and this is precisely, again, where people make discoveries. They don't discover anything that beyond what their sample are able to provide. Right, so you can also upload your own data that is, as long as it's compatible with the NHGRI EBI GWAS catalog data format. So you can upload your data, and then you would do exactly the same thing here. So I have loaded just now a preloaded data set of breast cancer. You also have, in fact, only 3 data sets have preloaded. These data sets can be downloaded directly from the catalog, but you can also choose one of the 3 preloaded data sets to go explore these things. So that's of very specific interest, I understand. I want to talk about next how I built this app. I think that will be of slightly more general interest. And if you want to do something like this, how can you make this yourself? I didn't know anything about front-end web development about a year ago, so I did this about in last February to March. And back then I had zero idea of front-end development, how to do interactive, visualizations, etc. I'm sure the audience must have more experience with these kind of things. But this tool is built with RShiny. So Shiny is a package in R that allows you to build interactive dashboards to visualize data. So basically a layer above R that runs in a browser that allows you to interact with the explored data. Now the basic structure of Shiny is that you have 3 files. You have your UI layer, so the user interface that specifies how you want to look at the data, what kind of panels you want to see in the browser. For example, I can specify there's a panel here that requests the inputs of model specification, and the output should go on the right, for example. And then there's the server, the second file, which is like your usual R or Python script. that tells you how to process the data. So once you take the data from the UI layer, you pass it to the server. Do your usual calculation. This is not really too far from regular script, R or Python script. Now you have your global file, which you can stash with your user-defined functions and any package loading operations in it. And then there's a I think the biggest advantage in terms of doing these kind of interactive stuff is you have talking between the exploration and the calculations underneath. Another advantage, they have dynamic UI. So if you want to do different things, for example, if you want to design your study, then depending on the input, you can have different outputs in your user interface. So this is something I think really nice. And it takes a little bit of time to build this, but it saves you much time further down the pipeline if you want to explore the data. So that's kind of the cool thing about these this Shiny app. And then you have data input and output, like I showed you just now. You can dynamically plug in your data set. It doesn't have to be preloaded data. It doesn't have to be hard-coded in. And then all these are achieved with something called reactive programming. So usually you have your object-oriented programming, procedural, imperative programming, but this is something related to the reactive nature of front-end programming. It's called reactive programming. So observe that once I click something here, the object output here will be different. So how does it know to change the output to visualize different things? Basically, what this is doing is underneath in the server part of it, in the script, it's observing what the front end is doing. So once something changes in the front end, it will try to update whatever is in the back end and then throw that back to the user interface. So you can kind of do that pretty easily, very easily. In fact, in Shiny apps, this is all packaged up. Now, it helps if you know a little bit of web development, especially reactive programming, but you don't really have to. You can build this pretty quickly in a month or in a week even. And there are plenty of examples floating out there. And outside Shiny, I would also like to advertise some other tools that I think work very well in this project. The first is Plotly. I'm not associated with them, but I also advertise them. So look at this plot here. All these plots are in fact done with Plotly. So Plotly is a company as well, but they provide some pretty fantastic interactive visualizations. So you don't have to do all these programming specifying that, let's say, if I hover my mouse here, it's going to show me some information about this particular data point. So this is all taken care of by Plotly. In addition, this Plotly also plays very well for interactive display. So just now we saw that when I click on a particular data point, the server is able to know that I clicked on a particular data point, update the information displayed here, and the plot as well. And that's because this Plotly allows you to capture whatever event that's happening inside the plot, inside the figure, so you can pass that back to the server. Now, the second little add-on I want to introduce is this intro.js. I'm only listing the names. I think each of these deserves a little bit of discussion on their own, but I'm only introducing the names of these tools. So this intro.js is a little introductory applet developed in JavaScript. So if you want to build an app and allow users to quickly explore what the app is capable of doing, you plug this intro.js into your app and just type in whatever you want to display at particular modules of this app. And then when you launch this little intro.js, it's going to guide the users through the interface or whatever you want to guide them over, and tell them what the app is capable of doing, and then it's a very nice guided tour that that otherwise a user will be overwhelmed with the interface of your app. So if you want to guide them through a little bit easier, this is something I find very useful. Now the final little add-on I want to tell you about is this load spinner. So when you load a web page, for example, when it's loading, you see a little spinner spinning around. Like, so it actually makes a difference if you didn't notice. So I think the server is responsive enough. You don't see this, but if let's say the internet connection is slow, there we go. Okay, so if the internet connection is slow and the picture hangs, you don't know what happens. This is something that tells you it's still going on. This is a very small point, but when you take this away, and I can give you maybe a demo offline on how this would look if you don't have the spinner. This will frustrate you if you don't know what's going on. Right, so that's everything I want to talk about this for this app and how I build this. You can there are equivalent tools in Python as well. There's a Flask framework for building web applications for data visualization like this. But I think it takes, I'm biased of course, but it takes a slightly it's slightly more difficult, and it takes slightly longer for me, at least, to build these kind of apps in Flask Python. And I think so far I find Shiny the best tool to build these kind of tools. So any questions so far? Yes.
How do you enter in data for GWAS studies that you have populating?
Sorry, can you repeat the question?
Oh yeah. How do you basically populate your tool with, let's say, new GWAS studies?
I see. So this is so the question is, how do I populate the app with the actual data from GWAS studies? So all these data are pulled in. So I'm going to talk about how everything is done from the beginning. So the data is downloaded from this EBI GWAS catalog manually. Okay, so I don't have an interface yet for directly requesting data from the GWAS database, but you can manually download this, and it doesn't take very long. Actually, it's just one click. So if you want the link, you can choose upload my data here, and it will give you the link to the header information on the EBI GWAS catalog here. So this will tell you the header information needed, the headers needed in that particular data file. For example, the date added to the app. So some of them are essential to uploading the data to the app. Some of them are not. Okay. And if you don't have the essential columns in your data set, this app will automatically detect that and tell you which one of the columns you need. Okay, so if you miss any columns or misspell any of the column names, it will give you a warning and stop you. But the easiest thing to do here is to download data directly in their data format. If you go to GWAS catalog, EBI GWAS catalog, and you go to download, you search for example, breast carcinoma, and you go here and you download. That's it. This download from their website is precisely in their own format. You don't have to do any formatting. But if you want to upload your own data, you have to go through the formatting according to the columns. But the thing with downloading from their catalog is takes quite a bit of time. So it can be quite slow, like you see here. So that's why I didn't integrate this into the app itself. Otherwise, it just hangs there for 5 minutes and not very productive. I'd rather, okay, yes. They have a tool for that. You should use their tool for this. Yes. So that's for exploring whatever is available in the literature, but you can also design your study according to whatever you need. So if you have a fixed number of cases, for example, if you want to study a particular condition, you have a certain number of people with that condition, you want to know how many controls you have to recruit in the population, you can go through this second tab, which tells you how many controls you need. For example, in this case, it's 6,000, whatever. But there's a third tab that does the disease model conversion. So this question that was asked is, so if you happen to have precise information about the biology of this particular disease, if you want to get at the two canonical, what I call canonical parameters, the risk value frequencies and the odds ratios, you can go through the third calculator, which converts the disease models into the canonical parameters. And you can directly copy. So let's say I have an additive model with disease prevalence 0.1, risk value frequency in the population 0.3, and relative risk 1.5. And this gives me 0.29 risk value frequency in control group, and the odds ratio about 1.5 as well. And you can copy directly by clicking here and go to this power calculator. So this number here, 0.29 and 0.15, is copied directly from that app to this. So yeah, that's another thing that you can do with this. Yes.
So in that one, what genotype are you referring to? The additive model. You just said genotype, relative risk. Are you referring to one or two alleles? Good question. So the question is, what genotype am I referring to in this model? Yeah.
In this box, right? Mm-hmm. So I'm assuming an additive model. So by the way, all these information in the slides is available as manuals in this app as well. So if you have additive model, genotype relative risk, like I specified, is 1.5 here. So the probability is 1 to 1.5 to about 2. So yeah, that's another good point you can make, is that the GRR numbers really mean different things in different models, right? So depending on what model it is, the third column and even the second column can be different, even if you have the same GRR number. So that is really model dependent. So the advantage of not going through this is that you don't have to know precisely. And another point you can make here is that different disease models can in fact give you exactly the same risk value frequency and odds ratio. So it's not a one-to-one correspondence. Multiple disease models can map to the same canonical parameter. For example, okay, I have an example here, in fact, down here. So down here, this is pretty complete since so many people have asked questions about this, is that if you have, let's say, a multiplicative model that gives you these two canonical parameters, you can have an additive model with slightly different disease model parameters that gives you exactly the same canonical parameters. You see, these are example of 4 different disease models that give you exactly the same canonical parameters. So this is not uniquely identified from data in a way. So, and that's, I think, a very good reason why people don't report the disease models in the literature, because it's not uniquely identified if you only have the 2x2 counts. That's a yes.
How much does uncertainty in your parameter estimates matter?
Right.
With regards to your position and the.
Right.
But since you have a phase transition there,
yes.
And how do you know that's like greater than the uncertainty in this?
Right. That's very good. So the question is about the uncertainty in the point estimates, right? So I can tell you right away that nobody reports this. So there are 2 layers of problems with the numbers reported in GWAS literature. So if you think that this number is so precise, it's exactly predicted by the theory here, I have whatever power calculations here, it's not the case. So the problem with these estimates is that the discovery and the effect size estimates are done with the same set of data. So what I mean by that is that traditionally, okay, back in the days, we have type 1 and phase 1 and phase 2 studies, right? Have phase 1 for discovery and phase 2 for confirmatory. And you use the data in phase 2 to estimate the parameters in your model so that you don't get this selection bias from the first. Now things are getting messy. So people say that we should pull phase 1 and phase 2 together to increase power. So you get biased estimates of all these parameters that people report. In fact, all these reports, 99%, I cannot say 100 because I haven't seen every one of the papers reported there, but all the papers I have seen, they report the numbers by using the data together. So I'm saying that I discovered these effects with this set of data and also estimated the parameters with the same set of data. So that creates a selection bias, right? If you have a large number of hypotheses to screen. So all these numbers are biased, actually upward biased, if you think about it. And there are no uncertainty estimates. There are usually uncertainty estimates associated with the odds ratios. I haven't plotted them. But to begin with, these numbers are not right because you have selection bias. But if you have unbiased estimates with uncertainty estimates, you can certainly plot them. Let's say so this wouldn't be a point. This would be a dagger, right, with uncertainty estimates around it. But if you have large samples, in a way, this and if the odds ratios and the error frequency are in the middle region, so if the risk error frequency are in the middle region, then if your estimates are not too far off, the horizontal part of the dagger will be small. But because this is kind of in a log scale, when you go into the tail part, when you go into the region where you're closer to 0 or 1, this dagger will be dragged quite a bit longer. So you have quite a bit uncertainty when you have really rare conditions, really rare genetic variants So I haven't done that. So, but the reason why I haven't done that is because it's not really possible with what's reported in the GWAS catalog. And we very much advocate for this. We go back to the phase 1 and phase 2 study and then try to come up with unbiased estimates. I think there's too much of an emphasis on discovery and not estimation. In my opinion, estimation is sometimes a more important question than reporting that this set of genetic locations are relevant. Yes, but thank you for the question. I think we have about 10 minutes. I won't go into the third part, which is the phase transition. So we saw that there's a pretty sharp transition between where you have power and where you don't. And there is a mathematical reason behind this sharp phase transition. Why suddenly you get power 1 from power 0 when you just increase the sample size by 200. There's a mathematical reason behind that as well. Yes, but that's about it. Any questions? Yes. Sorry, no. Okay. Yes. Yes.
So you said whichever way you kind of calculate, or separate method used to calculate power, they're all asymptotically similar. But which one did you use for this?
Yes, great question. So, so in the very beginning, I kind of talked about the question that when you perform different tests, you have different power, and these powers are asymptotically. The power of all these tests are asymptotic equivalent. Now, what I visualized is the common limit of all the power limits. So there's one. So this phase transition, this power calculation. Let's say if you have this many cases and controls, and if your underlying risk error frequency and the odds ratio is this much, then this power calculation applies to all these association tests, regardless of which one you choose. So this is the common limit. So if you go to, for example, Gonzalo's calculator, it'll give you pretty much the same answer, off by maybe 0.1%, that's it. But so you're safe using his as well, using any other calculators, but it's not quite clear. You see, so the biggest advantage I would say is to explicitly spell out which numbers you have to input into these kind of calculators. And the test part doesn't really matter.
So sorry, so which one do you use different tests based on different inputs, or?
No. It's the so the question is which one of the tests did I implement the calculations for? It's all of them at the same time, because the power limits is common to all these tests I listed here. So it doesn't really matter what kind of test you use, with the exception, okay, of when you have other controls. For example, when you do logic, this only applies to marginal screenings. So this is applicable when you don't have other controls. For example, if you want to do logistic regression, but you have other variables in there, then this doesn't strictly apply. But in the literature, what I found is that this applies pretty well. So even if you have other controls, it doesn't increase your power by very much, by an order of magnitude. It doesn't. So all these studies, one way or the other, use some kind of control, but it doesn't throw off the power approximations by orders of magnitudes. So that's the point I want to make. So this is applicable to pretty much all association studies, whichever you use. Yes?
Dotted red line mean?
Oh, dotted line is the accurate power curve. Thank you. Good question. Okay, I'll spend 2 more minutes on explaining what everything is. So the dotted line here is the accurate power curve. So what this means is that at this sample size, all the LDO frequency and odds ratio combination on the same line will give you the same power. So if you move along this line, you would have the same power of discovery. Now there's a red line here. It says rare variant zone. Okay. So typically, what people define rare variant is that they take a percentage, right? If you have a genetic variant that is present in less than 0.5% of the population, you call that a rare variant. So that's the typical definition of rare variant in the genetics literature. But I find that definition a bit problematic, because what are you using rare variants for? So you typically what people do is that if you have a rare variant, you know that your usual association test doesn't apply, right? Because it has distorted power. You don't have proper type 1 error control. So that's the concern there. So if that's your goal, your definition of rare variant should really be something different. It shouldn't be a percentage of the population that has a certain genetic variant. It should be the number of genetic variant present in the population such that you can properly control for type 1 error. You see, if that's the goal, you should target for that. So this is the number that calculates how many genetic variants need to be present in your study in order for you to properly control type 1 error. So that's a different definition of rare variant, and I'm very much in favor of this definition of mine. I'm biased, of course, but if you think about it, if your goal is to use the definition of rare variant to tell you when to use a test to properly control for type 1 error, then this is the way to go. But of course, I'm not that narcissistic. I implemented another version as well. So if you want to be traditional, if you want to say that I still want to define this as a fraction of the total population in the study, you can still do that. Right? So there's an option to specify the rare variant as a fraction of the total subjects in the study, and if you do that as typically done at 0.5%, this curve shifts, right, accordingly. Right, so in this case it's 61. But you see, this is not compatible with the power calculations here. So this danger zone, what I call danger zone, is really around here, not as compared to when you specify it as a fraction of the total population. And the good thing, one last, let me illustrate. So this definition is adaptive to the sample size in a way that if you have, let's say, this many cases and this many controls, you see that the curve shifts accordingly, marks the danger zone accordingly, as opposed to the usual definition. It's still not, it's never quite compatible in a way with the power calculations. Maybe the other way is more obvious. So you have more controls than you have cases. Then you see this is really far off if you define it as a fraction, but if you define it in this case, if you define this as the number of counts you need to control type 1 error, it doesn't even appear. It's never a problem for you if you have this many cases and this many controls. It's just a different concept. I think rare variant is a different concept in power calculations. And you can go through all these in here. So I have a user guide, pretty detailed with animated GIFs that tells you what each of these tabs do. I think I also have a pretty detailed document on what this rare variant business is. No. I'm also struggling to find it. Oh yes. So here you have a full supplement. Here you can read all the mathematics and all the definitions of rare variant in this document.
Okay, so I think maybe we'll stop there. We can thank our speaker.